I'm Michael Rugg. I'm the director of the UT Dallas Center for Vital Longevity. And it's my great pleasure to welcome you to what I think I am right in saying is the 10th Gene and Bill Busiotis Distinguished Lecture. It doesn't seem like the 10th, but time flies. And so this is a particularly special occasion, I think, in light of that. Support for this public lecture series comes from a bequest to our center that was made by Bill Busiotis in memory of his wife, Jean. And Bill was a tireless supporter of both the center and the university more generally. It's particularly fitting that the lecture should be in this space because Bill designed the building. He was the architect. And so we love coming back here. It helps us to rekindle our very, very fond memories of Bill and all that he did for our center and did for the university. And it's particularly gratifying to be able to welcome members of the Busiotis family to tonight's event also. And I'd like to give a special welcome to members of our Center's Advisory Council and Directors Research Circle. For those of you who don't know what the Directors Research Circle is, there is information skillfully planted around the building. You can pick up and learn about it. And anyone who's interested in joining us and joining the Directors Research Circle, please feel free to ask anyone who's associated with the center, particularly me. I will stay around if necessary, and you would be very welcome to join us and join our community. So the center's mission is cognitive health for life. And cognitive health for life means not only helping people to age as gracefully and as sharp, with as much sharpness as possible. It also means trying to figure out how we can help people stay as cognitively sharp and as cognitively healthy as possible throughout their entire lives. Not just for the last part of that, but across the entire lifespan. It's in that spirit that the talk tonight is of particular interest to us in the center. As I say, if anyone is interested in knowing more about what we do and joining our efforts, we would be more than happy to talk to you. Let me now turn things over to Dr. Joe Pancreaso, the Vice President for Research and Innovation at UT Dallas. He's going to make some remarks on behalf of the university. And Joe, thank you for being here tonight, and over to you.
When I was younger, I would just try to jump up this area, so I think is a measure of how I've aged cognitively in a good way. I'm not going to do that. So it's my pleasure to be able to welcome you all tonight. And thank you, Professor Rugg, for the opportunity to come and speak briefly, and I'll be brief, even though I'm a professor of bioengineering, and you're always at risk whenever you put a professor at a podium. I'm really delighted to give and welcome Ralph to Adolphs, who's the Bren Professor of Psychology, Neuroscience, and Biology. It's terrific to have you here. And, as a Vice President of Research and Innovation here at UT Dallas, I'm really delighted to be able to serve, and I mean that, serve the faculty, students, and staff as we pursue high-impact areas of research and address some of society's largest problems. And the range of what we do is large, from semiconductor energy storage solutions all the way to the cognitive function with aging and everything in between, astrophysics to zeolites, A to Z. Now, many of you know that UT Dallas was the inspiration of the founders of Texas Instruments just over 50 years ago. They envisioned an institution that was capable of producing the intellectual capital and the workforce that was necessary to sustain and grow what has become a technology economy in North Texas. Now, we're living in interesting times. What we're witnessing now is an exciting expansion to the breadth of that technology economy to include the life sciences. Evidence for this includes the success of Pegasus Park, nearby here near UT Southwestern, the recent opening of our Texas Instruments Biomedical Engineering and Sciences building, which is a joint venture between UT Dallas and UT Southwestern. It only took 50 years for that to happen, but we got there. And Dallas becoming the new ARPA-H customer experience hub. We're having a federal agency here concerned with actually transitioning biomedical technologies and bringing them to the public. So, these initiatives mirror what has happened in our rapid growth and our success in the biomedical sciences at UT Dallas, in fact. It's now one of the largest aspects of the portfolio that we have in research, which is extremely expansive. We even include clinical trials. So I think our founders would be amazed at the breadth with this tiny little school that they envisioned that was going to provide engineers and business people to drive, Texas Instruments and related industries, what it's become and where it can actually lead. So central to our research success is our own Center for Vital Longevity. And Professor Rugg touched on this very clear and compelling mission. It's to better understand aging and affects the brain and the mind, and it extends now to the entire lifespan, which is very exciting. That mission reaches and touches not only the center and the university, but it brings great value to our community. And that community, defined large, it's the public, engaged citizens, but it's also the scientific community around the world. The center scientists, through their own research and far-reaching collaborations, are really hoping to show us a path to be able to maximize our cognitive health over an entire lifespan. So on behalf of UT Dallas leadership, we are immensely proud of this center. We're delighted that Ralph has joined us today for what is the 10th anniversary. I'm sure we can get commemorative coins or something like that, I hope. And it's an honor for me to be able to count, my colleagues at the Center of Vital Longevity as faculty colleagues, and I mean that in all sincerity. So at this point, I'm going to turn the podium back over to Professor Rugg, and we'll introduce our special guest. Have a terrific evening. Thank you.
Thanks very much, Joe. So it is a great pleasure to welcome tonight's speaker, Dr. Ralph Adolphs. Ralph and I, we have known each other for, I'm guessing, more than 20 years, probably. He, as Joe just mentioned, is the Brem Professor of Psychology, Neuroscience, and Biology at Caltech. He's renowned for his research into the neural underpinnings of our ability to recognize emotional face expressions, into social behavior more generally, and individual differences in social behavior, including in people with autism. He is a pioneer of what is now the very extensive field of social neuroscience. He's received numerous accolades and awards for his research, including a Distinguished Investigator Award from the Social and Affective Neuroscience Society. The title for tonight's lecture is Social Cognition, Autism, and the Human Brain. Please welcome me in joining Ralph Adolphs. And welcome to Dallas.
Thank you very much, Mick, for that kind of introduction. I had the brief urge to try to jump up on the stage, but resisted. Okay, I think we're there. Okay, thanks again, Mick, for the introduction. Thanks to the Buziotis family for endowing this lecture series and to everybody involved in making this happen. And thanks to all of you for coming. So I'm going to spend the next 45 minutes or so telling you about work that we've been doing, trying to understand what we're doing right now, or what you're doing right now, and looking at faces, trying to understand people, trying to form a representation of what's going on inside another person's mind from looking at their behavior. And I'm going to anchor it on the clinical population that my lab has studied the most, which is autism. Okay, so, sorry, it's a little hard for me to see all these windows here. So this is the menu. I'm going to begin sort of broadly sketching the question and introducing you to how you might go about asking questions about what is different in the brains of autistic people versus people who are not autistic, and then turn to two particular processes that we've studied a lot, which is social cognition. That's the set of abilities I was just alluding to, how we look at people, think about people, and attribute to them feelings, emotions, intentions, and everything else that we think is going on in their minds. And then tell you a little bit, actually a number of things, about where we're going with this in terms of future directions. So as you probably know, autism, which was more or less contemporaneously conceptualized by Leo Kanner and Hans Asperger in the 1940s, is a pervasive developmental disorder, which means that it arises early in development, and most of the research on autism focuses on infants, children, because it is a developmental disorder and people are interested in understanding how it emerges in development. The population we study exclusively in my lab are adults, high-functioning adults with autism, for a number of reasons. One is just practical, that a lot of the studies require lying still in an MRI scanner, doing complex cognitive tasks, and so forth. But it's also relevant because, as the other word here says, it's not just a developmental disorder, it's a pervasive developmental disorder. So the babies and children that go on to develop autism turn into adults and grow old, and they still have autism. So it's a lifelong disorder that is characterized by a broad array of differences from people who don't have autism, some of which are itemized down here, and they keep changing when the Diagnostic and Statistical Manual of the APA comes out in various iterations. But one key difference is differences in social behavior, difficulty doing what I was alluding to before that we're all doing right now, difficulties in thinking about other people, connecting with other people, thinking about what's going on in other people's minds, and then more variably also delayed onset of language, a focus on detail, repetitive behaviors. So it's a constellation of a number of differences that are part of the diagnosis for autism. And of course, as the name says up here now, and everybody acknowledges nowadays, it is truly a spectrum. So it's a very broad range from people who are truly very disabled and mentally retarded to people who have exceptional abilities and everybody else in between. That makes it a challenge to understand. And so how might you go about understanding or investigating what goes on in the brain of an autistic person? I just wanted to give you a flavor for how this might look. So this is the Caltech Brain Imaging Center that Mick has visited. Here we would be scanning a participant's brain while they are engaged in a certain task. And this method, magnetic resonance imaging, would give you detailed pictures of a person's brain, like what you see here, person's brain, spinal cord, vertebrae, inside their skull and the rest of their body. And in addition, it would show us which parts of the brain are working particularly hard when they are doing something. And so what I will show you next is a video that it's real data, but it's a visualization of what happens in the brain of a group of people who have autism and a group of controls who do not. That's typically how we set up the experiments, when they're watching a movie that has a lot of these kinds of stimuli and would engage the processes that I was mentioning. It's full of people, and you're always trying to figure out what's going on in people's minds. So this is what it might look like. I had a person in my lab who was very fond of this sitcom, The Office, that you may be familiar with. So that's what we used as a stimulus. It's a good stimulus because it's full of people. It's people interacting, lots of funny and awkward situations. So people lie in the scanner and they watch this video, and you can see that this pattern of activation is changing over time. So we have a group of control participants here who are one at a time watching this movie, and we average the activity in their brain. So their brain's responding to watching and listening to this episode of The Office, and so is a group of autistic participants. Now the question you might ask is, well, what's the difference in the brain? And the place you might start is the visual cortex. So what I'm plotting here, it's a little dry data, but let me just tell you what this shows. Here on the horizontal axis is time while people are watching this movie, and we're measuring, that's plotted on the vertical axis, the activity in the part of your brain that receives visual input, occipital cortex at the back of the head. Each blue line is one control participant. So in some places there's something bright in the movie and activity goes up. In other places it's dark and dim and kind of boring and activity goes down, depending on the visual input. The average of this group is this dark line here. So you can see right away there's some patterns because everybody's watching the same movie. On average, some people's visual cortices are activated when it's something bright and lots of action happening in the video, and not so much down here. But there's a lot of variability between different people. Everybody's slightly different. In the group with autism, it looks like this. Here, the red curves are the individuals with autism, and the dark line is the average for that group. And so if we compare these two groups, the controls, the people with autism watching the same movie, what we find, shown in the bottom here, is that their visual brains actually respond very similarly. The curves look very similar. This is what you would expect. They're seeing exactly the same thing. Their brains are getting the same input, watching the same video. And so in terms of just what they're seeing, it's the same. The question then is what happens next? So you don't just see this movie, but this video, but you interpret it and you start thinking about the people. You might find some parts funny, and I might not, or a person with autism might not. So those further judgments, those further processing steps, can we find a difference in the brains of autistic people when we go to a brain region that's involved in that kind of activity, in complex social inferences? And one brain region that's known for that is this structure here, which is in the front of the brain, medial prefrontal cortex. And I show you the same data here. Here's the group of the control participants. Here's the autistic ones. And you see that there's not much of a difference between the groups. The reason for that is that it's not just that the control participants are different from the autistic ones. Every single control participant is different from another control participants. So I might find some part of this video funny, and a person with autism might not, but you might not either. Everybody's different. So this makes an important point that has been a challenge in understanding what goes on in the brains, what mechanisms are responsible for the atypical social abilities of people with autism, which is huge heterogeneity. People with autism are heterogeneous, everybody is different, and people without autism are also all different. So given that variability, especially in social cognition, it's been a challenge how to investigate this. I wanted to emphasize this point, as I mentioned at the beginning. There's a broad range of abilities and disabilities in autism, and the people we study are high-functioning adults, and indeed, we haven't studied this person, but there are people who have exceptional abilities. This is a person who lives in England, who's an artist, Stephen Wiltshire, who's autistic, and you can look him up on the internet. He goes into a helicopter and flies around a city like Paris or Tokyo or New York, and just looks and watches the buildings go by. Then he goes back to his studio, and over the next week, he sketches everything that he saw, and it's perfectly accurate, including the locations of all the windows in the houses. So this is amazing. There's some kind of amazing photographic-like memory. So again, huge range of abilities and disabilities in people with autism, and this has made it challenging to identify the neural substrates. This very dry slide here that I will go over very quickly just shows a couple of the seminal research papers that make the same point, that in autism, the brains of people with autism are less similar to one another. They're quite different. Even across time in the same individual, brain responses are different. And so there's a lot of variability across individuals and within individuals. And one question then is, how should we think of this variability? Might there, for example, be subtypes of autism that we want to identify? Or are there particular outliers, particular individuals that are driving group differences? And this is, in fact, something that we have found in our research. So that's the sort of intro. Let me turn to the two processes that we focused on. One is social cognition and the other is attention. And there's several reasons that they're relevant, but one is that both patently contribute to social cognition and behavior in the real world. So right now, You require social cognition. You have to understand what I'm saying. You have to be able to watch me, and you're forming an impression of what I'm doing, what I'm thinking, and that's part of watching this talk. And you also have to have attention to the relevant features. So both of these processes, mentalizing, or it's sometimes called theory of mind, attributing what's going on in the mind of another person from looking at them, and visual attention are key aspects of social cognition, and they're two processes that are thought to be atypical in autism. There's another reason that they're important, which is, as I mentioned, autism is a developmental disorder, and so if you have a baby that has difficulty in mentalizing, for instance, in playing with other individuals or pretending to play, or has difficulty in attention and doesn't look, for example, at the mother's face typically, that would lead to a different trajectory along the development of that baby and contribute to what you see when that baby develops autism, which is typically diagnosed a little later, around age 3 or so. So both from a developmental perspective and from just ongoing processing, a lot of the contribution to the social difficulties that people with autism have comes from these two processes. So one thing you could do is plot what I showed you before. So people are watching this video, The Office, and what I've plotted here on the horizontal axis is how similarly their brains are to the average control group. In red are autism brains, and in blue are control participants. And you can see there's a big range. Both the controls differ, there's a big range, and people with autism also, their brains respond very similarly to the control group, and some are quite different. There's a relationship, a weak one, in the people with autism, the red dots here, between how their brains respond to the video, the horizontal axis, and what they can understand about the video. That's plotted on the vertical axis. This is % correct on a questionnaire we give them. After they've watched the video, they come out of the scanner, and we ask them about the characters and what was going on. Did you understand the story? So this is preliminary evidence that there's some kind of link already here, even despite this variability, or in fact because of it, depending on how atypically your brain responds to the video, that seems to be associated with your social understanding of what it is that you saw. We can be more specific. Same stimulus, so again, people are watching The Office, and what we've done here is to try to isolate a very specific feature in the video that's shown at the top here. What we did is have a bunch of people in our labs who do not have autism sit and watch the video and rate how awkward they found different parts of it. The Office, if you're familiar with it, has lots of social awkwardness. There are a lot of awkward moments. For some reason, people enjoy watching this. And you can rate this. So there's some episodes here that are very cringeworthy, these peaks, and some that are not, etc. And so you get these mean ratings where people agree, this is really awkward, this is not. We can then use this time course of awkwardness ratings across the movie and ask, well, what's happening in the brain at those points? So what you see down here with these colors is those are the regions of the brain that respond to awkwardness. So we've identified some brain regions that are activated whenever there's something awkward in the video, and what's shown here on the left are those brain regions when we query them with respect to whether there's a difference in autism versus controls. It's, again, very weak evidence. Down here below the dotted line, these brain regions, we can be sure, do not have a difference between autism and controls. But these ones here, which are the colored ones here, that are involved in social cognition, in mentalizing, have some weak evidence for a group difference. So again, just like the previous slide, there's evidence, it's weak, it's noisy, but it begins to tell a story that indeed there are particular brain regions, more than one, that are components of a network that's involved in social cognition that is indeed functioning atypically in people with autism. We made a better controlled study where rather than showing people a video, we would show them individual faces of people under two conditions. So in this particular experiment, if you were a participant, you would see one of these faces at a time. I'm showing you the collage of all of them that people saw over the duration of the experiment. And in one case, we would ask you which direction is the person looking in. a very basic superficial question. Are you looking left or right? And in another question, we might ask you, how's the person feeling? Or are they thinking hard? So in one condition, you're just judging something physical about the face that you see. And in the other one, you have to judge something about the mind of the person behind the face. And that contrast isolates to some extent this process that we're interested in, this mentalizing process of forming impressions of other people. When you do that, through a number of papers, not just from our lab, but also many others, there are several brain regions, all these different colored dots that are involved in that process. Some of the ones were the ones you saw in the previous slide, but there are additional ones as well. We can then query these regions and see if activation of these regions while people are doing this task is related to any particular variable of interest and might be different in people with autism. So this is work done by my former postdoc, now junior faculty at Queen's University in Canada, Anita Tusche. And what we found is, I'll show you in the next slide here. So we have two types of data. People lie in the scanner, they do this task of looking at faces and making social judgments that isolates this mentalizing process. That's one set of data. And then in the same person, we ask them, how many people do you know, how many friends do you have? That's what's shown in this slide here. This is the number of social contacts you have in real life. It's a measure called the social network index. And there's a big range here. These are 3 control samples, a discovery sample, a replication, 2 replication samples, and a sample of people with autism. Each of the little dots is 1 participant. In many of these studies now, because of the variability and the often weak effects, we try to get many participants, and we often try to find something initially in one group and then replicate it in a completely different sample. That's why we have 3 different control samples here. But what we found is schematized here, and I'll just tell you about this very quickly, that we can predict the number of social contacts that you have in the real world from the social network index from the brain activation when you're doing this task. So there's a relationship between being able to do that social inference and engaging your brain, and actually in the real world having a healthy big social network. And that's the case both in the controls and in the people with autism. And there is also a relationship between the brain activation, from the brain activation of the social inference task when you're looking at the face. We can also predict in the controls a measure of trait like autism variability, the autism quotient, and in the people with autism, scores on one of the research instruments that's used to characterize autism, the Autism Diagnostic Observation Schedule. So in this study, we had associations between this particular process in the brain regions activated when you make social inferences, and actual real-life metrics. So let me turn to the second one. It probably some of you may have thought about this already. Visual attention is obviously really important, and a lot of everything that I've told you would depend, of course, on how people look at our stimuli in the first place. If they're not looking at the faces, then you would see altered brain activation. So that we can measure using eye tracking, and in all, for instance, our neuroimaging studies when people lie in a scanner, we also have a little camera that measures their direction of gaze so that we know where they are looking. So same stimulus as before. Now we're returning to that video, the office. Each colored circle here is 1 participant in the group of controls on the left, in the group with autism on the right. And you can see that they are similar in many respects. If there's only one thing to pay attention to, like a face, both groups look at the face. There's a lot of scatter. Some look away, some return to the face. It depends on what else is on the screen. And you could spend a lot of time, everybody in my lab did spend a lot of time looking at this and asking ourselves, is there some systematic difference that we can tell between how people with autism and controls look at this stimulus? You can't tell by just looking at it. You need to do an analysis like the one shown here. And in this analysis, we can decompose this movie into all the different aspects, the features, properties of it that might attract your gaze. In this case, it's any regions that have high contrast. So we go through frame by frame, this is done in an automated way, through the movie, and find high contrast regions like the shirt sleeves of the person, the lights up here, etc. And we can do it for lots of other features, color, orientation. And we can then ask, is there a difference in how people's gaze is driven, how their visual attention is captured by contrast, as opposed to the semantic meaning of what people are saying or other kinds of features. When you do that, you get data like the one shown here. So here we're asking, on average, where people look throughout the entire duration of the video, how much is their gaze driven by contrast? How much are they attracted to just things that are bright and contrasty? The control group is in blue, the autism group is in red, and indeed there's a group difference. People with autism, their gaze tends to be driven more by high contrast in this video than is the case for controls. So here we have a specific feature that we could isolate from this eye tracking study and find a group difference. We can do that across many different features and build much more complicated models, and we can ask questions not just about the group difference, but about individuals. That's important because if you look at the autism participants here, it makes the point that I mentioned earlier, which is many of these people with autism look at these stimuli, at least in terms of contrast, the same way that the controls do. They're not different. There are a few, these outliers up here, that are quite different, and their gaze is really attracted to high contrast, and they're the ones that are statistically driving this group difference. So we would like to be able to take this apart, to isolate these individuals up here and query them more, to ask, are there subgroups? To do that, we would need to have a lot more data and a lot more participants. So everything that I've told you so far, we have to schedule people, they come into the lab, we put them into a scanner, we set them up with an eye tracker. It's very slow. And so one future direction that we're working on and we're about to go live with is to try to do this kind of eye tracking in a much larger sample. Let me just tell you about that study. It's predicated on this recent paper that was led by a postdoc of mine, Umit Kellis. And in that paper, what we did was to show people 2 episodes of The Office on different occasions. And we got the data that's shown down here. So each of these colored dots is one participant with autism in the autism group. And in blue on the right, this is one control participant. And what we're plotting on the vertical and on the horizontal axis is how much time they spend looking to faces, but to do 2 different episodes of The Office, episode A, episode B. And you can see that all these dots are lined up across the diagonal. This means that the propensity to look at a face or to look at high contrast is something that is stable over time within an individual, like personality. It's a trait. So people this person doesn't look at faces in episode A, they don't look at faces when tested a week later on episode B. In general, they just don't look at faces as much as these individuals up here. So given this, we have temporally stable eye tracking. Our goal is to try to do this in very large samples, to do it longitudinally, ideally over years, and to track people's eye tracking to complex stimuli like movies using the webcam on their computers or the selfie cam on their cell phone. And so we're just starting these studies, so I can't show you the data from them yet, but it's led by these 2 individuals here, Nayeon Kim, who's a postdoc, and Qianying Wu, who's a graduate student and funded by a bunch of people, including Google, who's sending us the Pixel phones for the eye tracking on the phones. But with this, we're hoping to, in the next month or so, do eye tracking on about 5,000 people over the internet, including participants from the Simons Foundation whose genetics are known. So we're hoping to build up a big database, much larger than anything that I've shown you here, that would allow us to potentially find and characterize subtypes of individuals with autism or to characterize outliers from eye tracking to social stimuli and to videos with much greater power than what we had in the past. I can show you one set, just very quickly, of pilot data here. At the top are the data that I just showed you previously, showing that across 2 different episodes of looking at The Office, individuals have temporally stable eye tracking patterns. So they seem to be stable over a week. That's all we had in that paper. Down here, it's also, I think, about a week difference. And here we're using the webcam on people's computer and a program called WebGazer have a larger sample of participants, but again, we find the same thing. So we're hopeful that this will work, that there will be stable patterns. We can use eye tracking over the internet. And of course, then one additional challenge is to characterize autism traits over the internet. Some of the participants will have a diagnosis of autism. Many will not, but we will use instruments like the autism quotient and the SRS to look at individual differences. So that's one direction that we're heading in. I wanted to turn now back to the brain and to a story that we've worked on for quite a long time, and just give you one sense of that story because it has a very unique type of data. So in addition to working with autistic participants, that's by far the largest clinical population that my lab studies, I have a long-standing collaboration with the University of Iowa, as well as other institutions, to look at other clinical populations, in particular people who have focal damage to parts of the brain. And one region that we focused on is the structure called the amygdala, which is, I'll show you where it is in the brain in just a second. Here, the advantage is if we have focal damage to a particular part of the brain, and we can see some similarities between patients who have that lesion and people who are autistic, it would strengthen the hypothesis that this region is important or necessary, plays a necessary role in autism. And the advantage is that we would have a bona fide lesion in the brain from a stroke or a tumor excision that would give us much more certainty that this region is really critically involved. So what's shown here is eye tracking data like I showed you before. In general, if you just show a face on a screen, it depends very much on the context and the task. People with autism look less at the eye region of the face. You need to get a lot of eye tracking data to show this, but the difference is shown here. Control participants look more at faces. People with autism tend not to do so. And you find a similar pattern in this patient whom we studied a lot, who has a focal lesion of the amygdala. And so across a number of papers that are cited here, we looked at eye tracking and we tried to make comparisons between amygdala lesion and autism. It's one component of many different brain regions that are involved in social cognition. So obviously I'm not going to go through all of these in detail, but I just want to give you the general picture. What we're whittling away at here are particular parts of the brain. You've seen a few of the players, some of these regions here on the lateral surface, now the amygdala down here, but there are quite a few that are involved in social cognition, and they're presumed to be connected to one another, and you could think of them as nodes in a distributed network. So the amygdala, it's a strange word, it's Greek, it stands for almond, and that's because this structure is almond-shaped in your brain, and it's located, if you take a cut like this through the brain, down kind of in from your ears, this red region here. And some time ago, I teamed up with David Amaral, who's one of the foremost researchers on autism at the Mind Institute at UC Davis, and we took a survey. This is an edited volume that, in addition to papers from us, included papers from many other people, chapters, of what are the consequences of having amygdala lesions. And there's been a lot of work certainly linking the amygdala to emotion and social cognition, and if you have lesions there, you have deficits in those processes. And it's also been linked to autism. If you have lesions of the amygdala, as in this patient SM whom we studied, you don't have autism. She doesn't meet criteria for a diagnosis of autism, but you have a model of some aspects, in particular visual attention and social cognition, that would contribute to autism. There are other studies here. I'm just going to flash two up very quickly. This is a recent study from the IBIS network and Joe Piven. Long-standing research, and they're not the only group, showing that the amygdala has an abnormal growth early on in childhood. So within the first year, between 6 and 12 months, the amygdala grows atypically in those individuals who later meet criteria for a diagnosis of autism. So a lot of the work, as I mentioned at the very beginning of the talk, on autism looks at very early development to try to find predictive markers before the baby yet can be diagnosed to have autism. Can we find something in the brain that would predict whether or not that individual will develop autism? And this plot on the left shows that is the case. People have also looked at cells, individual cells within the amygdala, and again have found that there are subtle differences if you take a look at typically developed individuals' cells in the amygdala and those in people with autism. This is from a post-mortem, from a study of post-mortem brains. So there's a whole literature linking the amygdala, this brain structure, and cells within it, to social cognition and visual attention, and in particular also linking it to abnormality in the development of autism. So what did we do? Just to quickly tell you the story, we've been collaborating with a number of places that do epilepsy surgery. These people have epilepsy, and in some cases, to localize where the seizures start, the surgeon has to implant electrodes into their brains. So they're not getting these because of our research. They're already, for a clinical reason, getting electrodes implanted into their brains, and then they're sitting in their hospital room for about a week or two. It turns out, fortuitously for us, that in many cases the epilepsy originates close to the amygdala. So the electrodes are put into the amygdala. Because of that, we can record the activity of single brain cells from the amygdala of these patients. So they're awake, they're sitting in their bed, while we can show them pictures. This is in collaboration for this particular study with Cedars-Sinai Medical Center, in particular Uli Ruteshauser, who's now a professor there, former postdoc of mine, and the neurosurgeon Adam Mamalak, as well as several students. Actually here, Brooks Fu is at UT Southwestern close by, continuing some of this work, single unit recordings. So this is the setup. You have a patient, they have electrodes in the amygdala, they're sitting in their bed, they have a big bandage on with cables coming out. We show them pictures on a screen, we can do eye tracking, and we can measure single cell responses in the amygdala. So it's a really unique opportunity to have sort of the most reductive, precise brain measure from a human participant. Epilepsy and autism have a high comorbidity. You have a higher proportion of autism in people who have epilepsy than people who don't have epilepsy. And so we sat around for a number of years and by sheer luck got two people with epilepsy whose recording locations are shown in red here, who also had autism. So that's the contrast. We have a group of people with epilepsy, all of them have epilepsy, otherwise they wouldn't have the electrodes in the brain in the first place. We have a group of people who have epilepsy but don't have autism, and then we have two people who have epilepsy and they have autism. And we can record the brain cells. I'm not going to go through the details, although what this summarizes is that to first order, just the basic responses, electrical responses of the cells in the amygdala are the same. So it's not that we can't find the cells or they're just really abnormal to begin with. So we can record single cells in the amygdala. And what we then did is we did a study where we showed these individuals, these patients, not whole faces, but tiny little pieces of faces, one at a time. So sometimes you would see a little bit of an eye or a mouth or a nose. And we would then ask, when you show these pieces of faces, how do these neurons respond? So we add up all the pieces of faces to which neurons in the amygdala respond well in the controls, and it looks like this. So here, these hot colors, you can imagine a face underneath here, eyes and mouth. So in controls, amygdala neurons are driven strongly by the eyes, which would be the same regions of the face, if you show a whole face, that control participants, again, people with epilepsy but not autism, would also look, that they find interesting and salient. And the mouth, less so. By contrast, so this is the control again on the right, the two individuals with autism, their amygdala neurons did not respond at all, in fact, responded less to eye region of faces than to other regions of the face. So here we had a correlate of single cells in the amygdala that mirrored some of the visual preferences that we would measure with eye tracking to faces. And our interpretation of this is that the amygdala has some kind of interest or importance or saliency map of what you would find interesting and where you would direct your eyes when you look at the world. And in individuals with autism, it's not the eyes, and therefore they don't look at the eyes, whereas in people without autism, the eyes are particularly salient. So this is one very nice link where we had sort of one of the most mechanistic stories between a measure in the brain and a behavioral measure, how these neurons respond to little pieces of faces and the actual propensity of people to direct their eyes onto faces. Okay, let me turn to oh, sorry, I clicked too many things here. A couple of things here to close with that are future directions, ongoing, and I think very exciting future directions. So I had this bewildering slide up before and made the point, there are many regions of the brain that are involved in social cognition, as you might imagine. There are many regions of the brain that are different in people with autism. All of these have been studied in great detail. They can't function on their own. If we just have the amygdala there, of course, it wouldn't do anything. It needs to be hooked up to the visual system. It has to connect to other brain regions. And so the way to think about all of these little pieces of the brain is that they're connected into networks. If they're connected into networks, there needs to be something that connects them to one another. And those are white matter fiber tracts in the brain that form connections between brain cells and between different regions of the brain. If you take a brain, here's an actual human brain, a photograph of a human brain, looking at it from the top, so it's a big wrinkly area on the outside, cerebral cortex. If I carved away this top region here, you could look sort of inside, and what you would see is something that is what looks like this here on the right. This is a structure called the corpus callosum. It's about 200 million connections that connect the left and the right cerebral hemispheres, the most massive white matter fiber tract in the human brain. So there's a huge connection in the brain. It's very interesting to look at the development of this. I have a person in my lab, Lynn Paul, who has an NIH study ongoing to look at babies who are born with agenesis of the corpus callosum. So one question was, what role does this structure play, and is there any link to autism? Again, if you look across the literature, there is a link, and so people have found that in people with autism, the development of this corpus callosum is atypical. But what we've studied, what Lynn Paul, who's the person whose name I just mentioned in my lab, has been studying in adults and also in infants, and in fact in utero with using ultrasound in pregnant mothers, are people who don't have a corpus callosum. They're born without it. You may have heard of split brain patients. Those are patients that used to be done. Roger Sperry, who used to be at Caltech, got the Nobel Prize for his work on this, that had a surgical cut of the corpus callosum to treat epilepsy. These are not those. These are people that are born, to begin with, without a corpus callosum. It never develops. This is a healthy, typical brain on the right. This is a technique called diffusion MRI. What it shows is the orientation of connections in the brain. You're looking down at a brain here. The red is the corpus callosum. They're fibers going left to right. On the right is the brain of a patient with agenesis of the corpus callosum, and you can see there's nothing. The left and the right hemispheres are not connected to one another. And what Lynn showed some time ago in this brain paper here was that about a third of otherwise high-functioning individuals with agenesis of the corpus callosum meet criteria for autism. So there's a very strong link between autism, and in particular the social affective component of autism, and failure to develop the corpus callosum, which is shown, what I just said is shown here. So we have a couple of brain structures that we've looked at. We've looked at the amygdala in some detail with the single unit recordings. There's the corpus callosum that serves to connect different parts of the brain. There's one last one that I wanted to tell you about, coming back to this collection of brain regions that are involved when you make social inferences about people. So when you look at a face and you form an opinion of what's going on in the person's mind, from fMRI, these are all activated, and as I showed you before, some of them are activated differentially in people with autism, but that doesn't tell us how essential they actually are. What we would want to know is, well, what happens if you have damage to one of these particular regions, one of these nodes of this distributed network? So again, what we focused on here was this particular region, the dorsomedial prefrontal cortex, the same region that I showed you at the very beginning of the talk. It's involved in making social attributions, in figuring out if something is awkward or socially interesting about a video, but that has great variability across people. We showed in a previous paper that variability in activating this region correlates with lots of individual differences related to social functioning. It's like the other regions that I showed you, activation of this region when you make social judgments correlates with how many people you know are there in your social network in everyday life. That's shown here. And what we did here in this next to last slide is to take this task where you look at a face and you have to make social judgments about the face. That is shown here on this vertical axis. That your performance on that task is shown on the vertical axis. So the vertical axis here plots your ability to make social judgments, and the x-axis here plots the activation of this brain region. All these different colored dots are patients who have lesions to different parts of this network, all these different colored regions that I showed you. And what you find is a correlation. So just to jump to the main conclusion from this slide, you can damage, let me just go back to the network here, you can damage different parts of this network, all these different colored regions. Those are the different lesion patients we sampled. And you cause variable impairments in people's ability to make these social judgments. And those are associated with reduced activation of this dorsomedial prefrontal region. If you actually have damage to the dorsomedial prefrontal cortex, these are the blue ones here, then you're really impaired. So our interpretation of this is there's a distributed network. There are many components that participate in social cognition, but some of them are more essential than others. So one of them, in particular dorsomedial prefrontal cortex, is really essential for the network. If you have damage there, the whole thing goes. To the extent that damage anywhere else causes impairments in social cognition, it does so in virtue of reducing activation in dorsomedial prefrontal cortex. So this is just to give you a flavor for one direction in which we're going, is to build towards a more satisfying causal model, that we know many regions in the brain are associated with social cognition and atypical in autism, but which are really critical. So these kinds of data are getting us to that. Let me summarize. So I told you a number of things, a lot of things. Social cognition and attention, two of these processes that are critical to function in everyday life and do what you're doing right now. They're atypical in autism, depend on particular brain structures. And I told you some of the components here, prefrontal cortex that we just talked about with respect to social cognition, visual attention from eye tracking with respect to the amygdala. These need to communicate with one another. I didn't go into too much detail here, but I told you that white matter tracts, that is connections, physical connections in the brain between these regions, like the corpus callosum, are critical. So it's absent in agenesis of the corpus callosum. And this is what we think is at least one story, one broad story to tell about what is atypical in autism. Autism is characterized by atypical connectivity between those parts of the brain that are important for processes like social cognition and attention. Okay, I want to acknowledge, I mentioned a bunch of people going through, but acknowledge my whole lab, who are here at the Chen Neuroscience building at Caltech, just taken a couple of weeks ago. Funding sources and various resources, websites, GitHub if you're into looking at code, and in particular, thank all of you for your social cognition and your attention.
Thanks very much, Ralph. So we have time for questions. We have volunteers here with handheld microphones. If anyone wants to ask a question, please raise your hand and someone will bring a microphone to you. So we have a question over here. I can see a hand raised. Could we get a microphone? I'll get out of your way.
The thing that has always mystified me is this isn't a male disease, yet I know of few females that have ever been afflicted with this. I have many friends whose children have autism, grandchildren, but I don't know of any female. Can you explain that difference to me? I've always been totally amazed.
Yeah, you're quite right about that difference. and in general, one reads numbers like autism is more prevalent in males than in females by a factor of 4 to 1, or perhaps more than that for high-functioning individuals. And most people are coming around to the view that it just seems that way because females with autism are just come to attention and are diagnosed less often, probably for a number of reasons, but one reason is that they may just present somewhat differently than males with autism. So the current view is you certainly do have autistic females, and there's almost certainly it's less common in females, but it's not as unusual as you might think. And the challenge is to try to have a broader diagnosis and be able to get a better handle on that number. In our own work, in the lab, we have maybe a quarter or so who are female. For this web-based study from SOFA, when we've put it out over the internet, we get almost 50-50 actually, but that's not from a verified diagnosis. That's people self-reporting that they have autism. So it's definitely work in progress. It's autism is very highly heritable. It runs in families, and it has a very strong genetic basis, not only, but lots of work looks at the genes. Some of that is probably related to the big sex difference. But again, the bottom line answer to your question is twofold, threefold. One is, yes, it's less common in females. Number two, it's not as uncommon as people tend to think. And number three, we have to work on a better diagnosis so that we can include females and get a more accurate read on that number. It's almost certainly not as rare as people used to think in females.
Okay. Next question. Yes, question just here.
Hello, thank you for your fascinating lecture. I had a surprisingly negative response to those obscure facial stimuli you use, which is the impetus of this question. If the amygdala, which we know is developing atypically in individuals with autism, it's also well known to be involved in affective processing and fear responses. To what extent do you think differences in social cognition might be driven by a fundamentally different affective response to social contact in individuals with autism?
It almost certainly plays a role, and it's a very astute observation. Those images do look kind of creepy. And as you pointed out, the amygdala, one other big, huge field is the amygdala and its role in fear, and Pavlovian fear conditioning, recognizing fear in faces, and so on. One comparison that's interesting to point out is we did do an eye tracking study where people have to fix it at the center of the, people have to fix it on a fixation cross, and then a face comes up, and we arrange it so that your gaze necessarily falls onto the eyes. That location of the eyes is where the fixation cross was. And we measure how quickly you move your eyes away, your gaze away from the eyes of a face. In the case of people with autism, they do tend to move their eyes away from, their gaze away from the eyes more rapidly than controls. Which would support the hypothesis is that they're not just disinterested in looking at the eyes, but they actually have an aversion. The patient with amygdala damage, whom I mentioned, does not have that. In her case, she's just not interested at the eyes, in looking at the eyes, but if you arrange it so that she's looking at the eyes, she keeps looking at the eyes. So there is a big difference there, and the mechanism, whether or not there's an active aversion to social engagement, to looking at faces, looking at eyes, or even engaging and forming a representation of what's going on in somebody's mind, versus just difficulty or disinterest, I think is a really important one. My guess is that that distinction will serve to fractionate some subtypes of autism.
Okay. Yeah, question here, please.
Has your work given you any insight into whether magnetic resonance will be a good or an effective way of treating autism?
Well, it doesn't treat anything, but it could help diagnose. So magnetic resonance imaging, like the scanner I showed you, as far as we know, unless you have metallic implants in your body, is safe and harmless and doesn't actually do anything to you, but it allows us to visualize your brain. So it is helpful, And some of the studies, like the one from Joe Piven that I mentioned, use it to image brains and be able to diagnose or predict something about autism. But it's not an intervention. It doesn't actually act on the brain. Other techniques, actually stimulating the brain or doing ultrasound stimulation, those would actually do something to your brain. MRI does not.
Okay.
Okay. Yeah, we can take one more question.
Thank you for that insightful and thought-provoking talk. My question is a dreaded two-part question, so I apologize in advance. And the first part of my question is sort of related to a comment that you made earlier about autism, being on this continuum of ability to disability. So I'm wondering kind of how you think about the way you study social cognition and if that's should be a target of treatment. And then if it should be a target of treatment, given what you found in the brain regions that you've investigated, them being both gray matter, white matter targets, if you think there's a specific brain region that may be more targetable or treatmentable?
Well, the brain region that, people are trying all kinds of places, including stimulating the amygdala. One might think dorsomedial prefrontal cortex would be a target. But to answer the first part of your question that kind of takes care of the rest, at least for the participants that we study, I don't think that treatment would be the right word or the right approach. And I think in general, I know from talking to them in general, they wouldn't think so either. They have difficulty to some extent in social interactions, but I think the treatment probably is applied best to the rest of us by educating us about autism and what it is, and providing suitable and structured environments so that high-functioning individuals with autism are able to function. I think, again, of course, there are some individuals who are mentally retarded, severely disabled, and their treatment makes more sense. But there's a broad spectrum, probably including Caltech professors, for example, where treatment doesn't seem like the right thing. And again, I think acknowledging that there's just huge individual differences, and then trying to find out more about autism and what characterizes autism, and educating everybody about that would be what I take away from it. But you had a second part, or were those the two parts?
You touched on both of them. Thank you
Okay.
so much.
Okay, I'm going to bring the formal proceedings to an end. Ralph, if you're willing to stay around for a little while afterwards, if people want
By all
to
means,
ask
yeah.
questions directly to you. Could you please join me again in thanking Dr. Adolphs for such a wonderful talk? I'd like to thank you all for being here on this momentous night for